So good morning all of you. Welcome back to another video lecture on cell biology. Today we are going to discuss about ribosome. As far as ribosome is concerned, we'll first go to the discovery, then distribution, then types, then structure. So first come to the discovery. Ribosomes were first observed in 1941 by Albert Cloud. Then in 1953, Robinson and Brown first discovered the ribosomes in plant cells. In 1955 rather, Palladay observed the ribosomes in animal cells and coined the term ribosome. So you can see that starting from 1941, in the year 1955, the term ribosome was first coined by Palladay after he observed them in animal cells. Now come to the distribution of ribosomes. Ribosomes are found both in the cytoplasm as well as in some of the organelles like your mitochondria, chloroplast, etc. So cytoplasmic ribosomes, they are either found freely or they may be found associated with some structures like rough endoplasmic reticulum. That is the reason why the endoplasmic reticulum has been divided into two categories, that is smooth and rough. So in case of smooth endoplasmic reticulum, there is absence of any ribosomes, whereas in case of your rough endoplasmic reticulum, there is presence of ribosomes, isn't it? So as far as types of ribosomes are concerned, ribosomes are basically divided into two categories, that is 70S ribosomes and 80S ribosomes. And these different types of ribosomes are divided on the basis of, ribosomes have been divided on the basis of sedimentation coefficient. We will discuss about sedimentation coefficient in another video lecture. So on the basis of sedimentation coefficient, ribosomes are of two types, 70S type and 80S types. The 70S ribosomes, they are found in prokaryotic cells and in your some of the organelles of eukaryotes like mitochondria and chloroplast. Okay, and the 80S ribosomes are found in only eukaryotic cells. So you can say that 70S ribosomes are confined to prokaryotes and some of the organelles of eukaryotes like mitochondria and chloroplast, but 80S ribosomes are exclusively found in eukaryotes. So now come to the structure of ribosomes. You know that ribosomes are membraneless, that is, are membranous. At the same time, ribosomes consist of two unequal subunits. One is a smaller subunit and one is a larger subunit. The smaller subunit is your ellipsoid in shape. So it is a small oblate or ellipsoid subunit, and the larger subunit is somewhat dome shaped. Now 70S ribosomes consists of two parts, 50S, which is the larger subunit, and the 30S, which is the smaller subunit. Similarly, the 80S ribosomes, these consist of again two units or subunits. What are those? 60S subunit and 40S subunits. Okay. So the 50S subunit in case of 70S ribosomes consists of 23S ribosomal RNA along with 5S ribosomal RNA and 34 proteins, around 34 ribosomal proteins. And the 30S subunit, it is made up of 16S ribosomal RNA and 21 ribosomal proteins. Okay, and you know very well that 70S ribosomes are found in prokaryotes and some of the your eukaryotic organelles like mitochondria and chloroplast. Whereas the 80S ribosome, which is exclusively found in eukaryotic cells, these are made up of two subunits, 60S and 40S. What are those? Your 60S subunit is made up of 28S ribosomal RNA, 5.8S ribosomal RNA, 5S ribosomal RNA, and around 40 ribosomal proteins. Then the 40S subunit is made up of 18S ribosomal RNA and around 36 ribosomal proteins. Okay. Now you are coming across the term or letter S. Well, as far as S is concerned, it is otherwise known as your Wedberg constant or Swedberg constant. Okay, we will discuss in details about it in another video lecture. What is Swedberg constant? Okay. As far as the two subunits are concerned, their association or their dissociation, that depends upon the magnesium ion concentration. Now, magnesium ion concentration determines whether the two subunits will get associated to form a dimer, or the two subunits will get dissociated into separate units. Now, increase in the magnesium ion concentration actually results in the formation of dimer. That is, when a magnesium ion concentration is high, the two subunits are joined to form a dimer. During protein synthesis, many ribosomes are arranged, got it, on a single mRNA. Now, to form this kind of structure is called polyribosome. So, polyribosome is nothing but during protein synthesis, the many association of many ribosomes on a single mRNA is called polyribosome. Now, let us come to the various parts of the ribosome. Now, as far as ribosomes are concerned, there are 4 sites, okay, which have got specific functions, got it, during protein synthesis. First one is A site, second one is P site, third one is E site, and the fourth one is your mRNA binding site. So A site is nothing but it is the amino acyl tRNA binding site. Okay. P site is the peptidyl tRNA binding site, and the E site is the exit site. What happens? The uncharged tRNA it comes to the E site, and then it leaves the ribosome and enters into the cytoplasm, re-enters into the cytoplasm. That you will study in details during protein synthesis. Got it? And last one is the mRNA binding site. So these are the 4 sites which have got specific role during protein synthesis in case of your ribosome. So functions, as far as function is concerned, very simple. The main function of ribosome is protein synthesis. Got it? Now you know that ribosomes are found both freely as well as they are also found to be associated with your endoplasmic reticulum. Now those ribosomes which are found freely, they actually synthesize proteins, and these proteins are meant to be used inside the cells. But those ribosomes which remain attached to the endoplasmic reticulum, they also synthesize proteins, but these proteins are meant to be transported out of the cell or into the plasma membrane. So basically you can say that the function of ribosome is protein synthesis. Now let us come to the diagram of ribosome. As you can see here, the ribosome consists of a smaller subunit and a larger subunit. Now this one is the larger subunit and this one is the smaller subunit. Got it? When these two larger and smaller subunits, they get associated. So as you can see in the diagram shows that there is presence of a larger subunit and a smaller subunit. The larger and smaller subunits, they get associated to form a dimer. This is the dimer. Okay. Under normal condition, the larger subunit and the smaller subunit, they remain separated from each other. And you know that when they get associated, when the magnesium ion concentration is very high. So you can see that here in this diagram, this is the larger, this is the smaller subunit, and the 3 important sites of the ribosome that has got specific role during protein synthesis are written here. This is the A site, that is the amino acyl tRNA binding site. This is the P site, that is the peptidyl site, okay, peptidyl tRNA site, and the E site, this is the exit site. Got it? So these are the 4 compartments of the ribosome, and the 4th one is the mRNA binding site. This is the mRNA binding site. Got it? So this is what the structure of ribosome is. It's a very simple one. I hope the video will be helpful for you in understanding the structure of ribosome. Thank you.